Hello from San Diego, where, as you know, the 65th annual ASH meeting is underway. And this, for us MPN researchers, is a particularly exciting meeting. In fact, I don't recall an ASH meeting or any other meeting with as many exciting advances being presented as at this meeting. And so I'll just try to go over a few of them that I'm excited about. First of all, there are two phase 3 randomized clinical trials being presented at this ASH meeting for myelofibrosis. Both are investigating synergistic combinations with a ruxolitinib backbone. One investigates pelabresib, a BET inhibitor, and the other investigates navitoclax, a BCL2 and BCL-XL inhibitor. Ruxolitinib has been standard of care for most patients with myelofibrosis for many years now, and we are perhaps on the cusp of a paradigm shift if the combinations are to get approved. So these, like I said, are synergistic combinations. They are being tested in large phase 3 trials against placebo, with both arms receiving ruxolitinib, and those presentations are being made today, Sunday, December 10th, here in San Diego. There are a number of other exciting emerging novel agents that are being looked at, and data being presented from phase 1 and phase 2 trials from these agents. These are, of course, open label, single arm, non-randomized studies at this point, and there's quite a few that I've been involved in and I'm looking forward to. Some examples would be the ALK2 or ACVR1 inhibitor zilergisertib, that's a mouthful, from Incyte, which is a drug for anemia. As our listeners are likely aware, recently momelotinib was approved by the FDA for patients with myelofibrosis and anemia, and the way momelotinib improves anemia is by blocking ACVR1 or ALK2. So this drug, zilergisertib, does precisely that and could be a nice partner for ruxolitinib, which again has been the standard for so many years, has a survival benefit, and is undeniably a very valuable drug for our patients. So this might be a way to actually sustain the hemoglobin levels while on ruxolitinib and nicely complement its action. There are many other drugs being presented. There is a PIM kinase inhibitor from Sumitomo. It's called TP3654. This is an interesting agent, a novel agent, new mechanism of action. This is being studied on its own at this point. Early trials, they're looking at different doses. So that's something I'm certainly watching. And again, in the sort of in the same spirit of novel agents with different mechanisms of action, there are umbilical cord derived regulatory T cells from a company called Celenkos, which has a partnership with Incyte. This data is being presented as well, was presented last night at the poster session. These are these cells given to patients who are already on a stable dose of ruxolitinib. So a foray, if you will, into immunotherapy or cellular therapy in myelofibrosis, which would be a new, again, another paradigm shift, a sort of groundbreaking in that regard, since we've not had immunotherapy or cellular therapy that really has had any meaningful role to date. So this could be something exciting. There are again many other drugs that that are probably not possible to cover in the context of this short interview, but others that come to mind include selinexor. Now we've heard about this compound at prior meetings, so there's an update being presented at ASH. But selinexor is actually another rational combination that's going into a phase 3 study very soon, and we'll start to enroll. This would be a pivotal trial of selinexor and ruxolitinib versus ruxolitinib and placebo, just like some of the others that I said are being presented today. Turning to the other myeloproliferative neoplasms, again, as you know, most of the drug development action does tend to occur in myelofibrosis, but important to remember that there's also a lot happening in the other myeloproliferative neoplasms. For example, in polycythemia vera, where we've used ruxolitinib now for nearly 10 years, there is a presentation by Stefan Koschmeider from Germany about using ruxolitinib frontline in polycythemia vera patients, which is certainly intriguing and something I'm looking forward to hearing later today. Ruxolitinib, of course, to date is used after hydroxyurea resistance or intolerance. So this would be something pretty new in that regard. I presented yesterday results from the first results from a trial called Summit, and that looks at a highly selective and potent KIT D816V inhibitor, bezuclastinib, in patients with indolent and smoldering systemic mastocytosis. Early results are very encouraging. This is a highly active drug, and we look forward to further results from this trial as more patients are reported on sometime next year, and then to the randomized part 2, which will look at this drug versus placebo. Similarly, there were data presented on elenestinib, another potent and selective KIT D816V inhibitor that also, like bezuclastinib, does not cross the blood-brain barrier, and the hope is that these drugs would be able to deliver the same or higher efficacy than, say, we currently see without incurring the liabilities of central nervous system toxicities or hitting targets that are not meant to be hit, by virtue of them being so selective for the target and also not crossing into the brain. And probably I'll end with one that I found really intriguing in Saturday's oral session, which was about the risk of patients with indolent systemic mastocytosis developing advanced systemic mastocytosis. We have generally thought of this to be a rare occurrence, something that does not normally happen, and the general feeling has been that indolent SM patients enjoy a normal life expectancy, which is still true for the most part. However, there was a thought-provoking abstract presented at the oral session that showed that the risk of progression from indolent to advanced SM could be as high as 18%. Of course, data such as this needs to be validated in other studies, but certainly quite surprising for the field as a whole.